Good morning, children. Now the last part we have come in this chapter, that is called microorganisms involved in bioremediation. There are two types of microorganisms. We know that aerobic and anaerobic microbes. The aerobic microbes, when they are degrading the pollutants, they need the oxygen for their activity, for decomposition. So aerobic microbes degrade the pollutants in presence of oxygen. They mainly degrade the pesticides and hydrocarbons. One example and two examples have been given. One is called Pseudomonas putida. It's genetically engineered microorganisms that had been developed by the Indian-born American scientist, which is called Anand Mohan Chakrabarti. Obtained patent for this recombinant bacterial strains. It is a multiplasmid hydrocarbon degrading bacteria. Multiplasmid, generally one plasmid will be there. As it is artificially made, it contains so many plasmids which help in the degradation of the hydrocarbon, which is present in oil spill. That is that microorganism, genetically engineered microorganism, GEM it is called. Second example, Nitrosomonas europaea, maybe we have discussed in 10th standard also, is capable of degrading benzene and a variety of halogenated organic compounds, including what are the halogenated? Chlorine, bromine will be present, trichloroethylene and vinyl chloride, helping in degrading the halogenated substances, organic compounds. Next one, Ideonella, very important. Ideonella sakaiensis is currently tried for recycling of the PET plastic. These bacteria use the PETase enzyme, PETase, PETase enzyme, and PETase and methatase enzyme to break the PET plastics into plastics will be converted with the help of these two enzymes, will be converted into terephthalate, terephthalic acid or terephthalene and ethylene glycol. That is the importance of Ideonella sakaiensis. Now these are the three examples for aerobic microbes. Now anaerobic microbes. Anaerobic microbes degrade the pollutants in absence of oxygen. That is the meaning of this word. Examples: Dichloromonas aromatica. Dichloromonas aromatica has the ability to degrade the benzene anaerobically. Benzene can be degraded anaerobically and to oxidize toluene and xylene. Another one, Phanerochaete Chrysosporium, an anaerobic fungus, exhibit strong potential for bioremediation of pesticides, polyaromatic hydrocarbons, dyes, trinitrotoluene, then cyanides and carbon tetrachloride, etc. These chemical substances can be used for their growth, and these chemical substances can be reduced from the environment. Now next one, Dihalococcaides. Dihalococcaides species are responsible for anaerobic bioremediation of toxic trichloroethane to non-toxic ethane. So trichloroethane will be converted into non-toxic ethane. Next one, Pestalotiopsis. Pestalotiopsis also you can say, or Sciopsis microspora is a species of endophytic fungus. Species of endophytic fungus inside the plant's body. This fungus will be growing, capable of breaking down and digesting the polyurethane. This makes the fungus a potential candidate for bioremediation projects involving large quantities of plastics. So that this is the end of this topic. Okay, thank you children. Next day we will be discussing the question answer.